Hello, everyone. Welcome back to my NicheNet video tutorials. In this video, I'm going to show you how to analyze ligand-to-target signaling paths. So first we know the libraries NicheNet and also tidyverse. For the ligand-to-target signaling path analysis, we still need to know the prior model, weighted networks, ligand target matrix, and the ligand receptor network. In addition to those prior models, we also need to know the signaling networks and the gene regulatory network. So in video 2, we performed the NicheNet analysis, and we identified the target genes for top 20 ligand from monocyte NK cells and dendritic cells to fibroblast in IPF lung. So I saved the target genes as a CSV file for the top 20 ligands. So we can read this CSV file back. So you can see here we load the target gene back into R. So let's connect the data set and have a look at the target genes. So you can see the ligand column are the top 20 ligands, and the target column are the target genes for each ligand. In video 2, when we performed the NicheNet analysis, we identified IL-1B as the top 1 ligand to regulate gene expression in fibroblast. So we are going to use IL-1B as an example to show you how to analyze ligand-to-target signaling pathways. You can use any ligand that you are interested in. So you can see from row 1 to row 41 are the target genes for IL-1B. So we can just get the target genes for IL-1B. So we can select row 1 to row 41 as active IL-1B target links. So you can see now we have a data frame that contains 41 target genes for the IL-1B ligand. So let's have a look at the data set again. You can see now we only have 41 rows, and all the genes are IL-1B target. Because we are going to use the IL-1B ligand as an example, so we can set the ligand as IL-1B. You can also set more than one ligand if you want to analyze several ligands at the same time. So here, for demonstration, we just set the ligand as IL-1B. Then the target will be the 41 target genes I show you in this data frame. So we can set what target genes as target all. Because this is a demonstration tutorial, I ran this analysis before. If we set all the target genes here, it is almost impossible to look at the signaling pathways because we have too many target genes here. So for demonstration, we can just select two genes, BMP2 and PTGS2 as an example, to show you how to analyze ligand-to-target signaling pathways. So for the demonstration, I'm going to set the target all as BMP2 and PTGS2. So now we can get the ligand signaling path for IL-1B to target gene BMP2 and PTGS2. We need to use the ligand target matrix, and also we set the ligand as IL-1B and the target as BMP2 and PTGS2, and also we need the prior model weighted networks. So now we can get the ligand signaling path, and we named it as active signaling network. So let's run this code. So you can see we generated a data list as active signaling network. So let's connect the data list to have a look. You can see we have two lists here. One is the signaling network, and the other one is the gene regulatory network. Because we are going to show the signaling network and also the gene regulatory network in the same figure, so we need to normalize the weight for both networks, then we can plot them in the same figure. So in order to normalize the edge weight, we first set a new name as active signaling network minimum and maximum. Then we can normalize the weight for the signaling network. Next, we normalize the weight for the gene regulatory network. So now we can generate a graph to show the signaling pathways from IL-1B to BMP2 and PTGS2. And we can use the random graph function in the diagram R to view the graph. So the layout will be tree graph. So let's zoom in. We can make it bigger. You can see IL-1B signaling to target gene BMP2 and PTGS2. So from the analysis, we can clearly see three signaling pathways. One is through the NF-kB1 and REL1. So this is the NF-kappa B signaling pathway because both are the subunit of NF-kappa B signaling pathway to BMP2 and also to PTGS2. And the second pathway is the JNK pathway because MAPK kinase 8 is JNK kinase. Through JNK kinase to gene, then regulate BMP2 and PTGS2 expression. So the third signaling pathway is the STAT1 signaling pathway to regulate BMP2 and PTGS2 expression. And the PTGS2 also known as COX2. So from previous published research papers, we know those signaling pathways are important for fibroblast transition into myofibroblast in human lung. So here we just use the IL-1B ligand as an example and only showed the signaling pathways to BMP2 and PTGS2. So you can practice to see if you can run the analysis for IL-1B to target gene BMP2 and PTGS2. If you can run this, then you can also practice other ligand and different targets. So this is a very short video, but you can see through the NicheNet analysis, we can show important signaling pathways in IPF lung. So I hope this short video tutorial could help your signaling pathway analysis. I'm going to stop here. Please subscribe to my channel if you haven't to do so, and share my videos with your friends. Thank you and see you next time.